Okay, so today I'm going to talk to you about discovering circulating tumor cells. So in 2012, 13.7 million people were under the care or had a history of cancer. In 2014, it's estimated that approximately or greater than 1.65 million people will be diagnosed with cancer. Cancer is the second leading cause of death in the United States behind heart disease. But what is cancer? Cancer is a localized mass of malignant cells. These cells can start in a primary tumor. They are able to grow. Eventually, they break through the basement membrane, and they're able to enter the circulating tumor cell, the circulation. These circulating tumor cells then flow freely through the circulation. Eventually, at some point in time, they are able to adhere to the cell wall, break free, and start a new tumor or metastases. What is interesting is you have tumor cells floating through your blood. Basically, you have the ability to define and understand a tumor before it grows, before it gets somewhere else. So, what are the implications of identifying and understanding these circulating tumor cells? Well, if I can identify these circulating tumor cells in the system, I can detect cancer early in healthy patients. I can give a prognostic indicator for patients once you find these circulating tumor cells. I can give you what the outcome is going to be if we're able to characterize these cells. You can treat the primary tumor right away. I didn't even know I had a tumor, but yet somehow I found the circulating tumor cell, and yet I can then treat it because there'll be some characteristics known about this cell. I can interfere with the metastatic formation or the formation of new tumors. The idea here is you want to be able to locate, identify these circulating tumor cells, take them out of the system, and characterize them. The most important thing, I think, and the thing that I want to get across today in this field is that if you are able to identify and characterize these circulating tumor cells, you can personalize medicine. At the end, what I hope to show you is that people and a lot of time and energy and great scientists are working hard to develop techniques that will allow you to take blood out of a patient and isolate a living circulating tumor cell, which at that point in time, you can expand upon this cell because it's an immortalized cell that grows. You can take the cell and you could give it a battery of therapeutics. Basically, I found a tumor cell, oh my gosh, do I have a tumor? Well, maybe. Give you some therapeutics and potentially cure cancer before it even gets out into the system. Circulating tumor cells were originally discovered by Thomas Ashworth in 1869. He made a general observance that tumor cells escape into the blood or other tissues. Now you might ask, what's taken us 150 plus years or something like that to discover or develop techniques to uncover this? Well, tumor cells are rare. To start with, tumor cells, when they break through into the blood vessel, most of them die. Only a few remain. What happens is there's variation within the tumor. So you get a tumor mass that's growing, there's variation within the tumor. These cells are able to break free, they go into the blood system, and at that point in time, most die. A few remain. So there are about 1 to 10 milliliters, 1 to 10 circulating tumor cells in a mill of blood. Now you might say, oh, that's not so bad. However, in that same mill of blood, there are 5 billion red blood cells, 4 to 11 million white blood cells, and 200 to 500 million platelets. A 1 to 1 million ratio is equivalent of stacking a dime in a mile high of pennies. So these circulating tumor cells are extremely rare. In addition, the circulating tumor cells within the cells within the tumor have variation. Ooh, excuse me. Right? As the tumor grows from a single cell, you're going to have variation within the tumor. They're going to be larger and smaller. You're going to have different nucleus to cytoplasm ratios. And as they break free and enter the circulating tumor cells, what you're going to have is you're going to have different characteristics within these cells. And even the cells themselves change depending upon their environment. So what you have here is a needle in a haystack. You have circulating tumor cells that vary in their characteristics. You have a very low abundance. But you'd be amazed at the amount of progress that this field has taken. Essentially, to isolate and understand these circulating tumor cells and to characterize them, there are two basic approaches that I like to discuss. There's the enrich and isolate. You need to be able to get that circulating tumor cell away from all those other cells with which to characterize it. You then need to be able to detect and characterize it, meaning you need to be able to come up with a system that tells me that cell is really a circulating tumor cell. So, the interesting part is there are all sorts of strategies coming up, and people are designing and implementing all sorts of unique and wonderful strategies to enrich and isolate these circulating tumor cells. There is a system in place right now using fluorescently labeled antibodies, and we'll talk about that. But what I find really fascinating is this next generation sequencing, the next generation characterization of these circulating tumor cells. Sequencing, as you guys have heard with the Human Genome Project, understanding what the genetic makeup of these circulating tumor cells are is going to be very powerful. And more importantly, what they've really been able to do is they're going to be able to isolate live circulating tumor cells. And as you can imagine, these cells, once again, they proliferate. You can then use these proliferated cells and you can give it a bunch of different therapeutics to determine the best medicinal direction. So, research that I've worked on in order to enrich the circulating tumor cells included a filtration membrane. Basically, you take your pasta after it's cooked, you pour it into a colander. What happens? All the water goes through. Same principle. We take a filter. The platelets, the red blood cells, the lymphocytes, you can see they're all smaller than an 8-micron pore. Plus, as you would imagine, these cells are a little bit pliable and they can be pulled through. The larger neutrophils, macrophages, and circulating tumor cells, they can sit on top of the filter. As you can see from this image on the right, this is a large filter with a bunch of cells on it. The number of cells is not as great as the billions we're talking about with red blood cells, but at the same time, it's highly enriched circulating tumor cells sitting on this filter. As you see this more blown up picture, you can see the pores in blue, the pores in these dark black circles, and the cells are in blue. So, I use filtration in order to isolate these circulating tumor cells. And now, how did I detect these cells? Well, there's something called, there's something called antibodies. On each one of these cells that we have, there are specific characteristics. Now, I know I told you these characteristics change, but some of these characteristics are pretty constant throughout most of these cells. So for instance, and I'm giving a schematic here, white blood cells, defined by WBC, they have blue squares on them. Most of them will have blue squares. Circulating tumor cells have red triangles. So these cells have specific markers on them. You can engineer antibodies specific to find and recognize these markers. So if I put an antibody specific for my blue square in, it will recognize and bind to the white blood cell. If I put an antibody specific to the red triangles, it will bind and identify circulating tumor cells. Why is that important? Because now imagine if I put color on it. Imagine if I'm able to put some sort of color that allows me to identify where the blue marker is. And imagine if I can put a green color on the red triangles. I can put a green color on the antibody, which recognizes the red triangles. At this point in time, I can detect where the white blood cells are, and I can detect where the circulating tumor cells are. So my strategy was to enrich and isolate using filtration, and then to detect using fluorescently labeled antibodies, and, if I was lucky, using some sort of sequencing. Take that cell off and sequence it to determine what is the DNA sequence of some of these known SNPs or other areas that are known to be problematic. So, as you can see here, when I take the filter, and in that one area, and I know you'll say, well, how did you get there? We use a microscope. So, using this 4x image, then blown out into a 40x image, you can see in that one particular area is this circulating tumor cell. When it's blown up, or when the filter channels are separated, and we use a stain specifically to identify the nucleus of a cell, you can see that is a cell. You can also see, using my my antibodies, which locate my red triangles on circulating tumor cells, I can definitively say that is a circulating tumor cell. In addition, I can also say this is definitely not a white blood cell. The reason why? I have an antibody specific for antibody labeled with an orange color that specifically labels my blue squares found on white blood cells. Now, we then started to use this on patients with prostate cancer. And here is an example of what we were able to do. You can see when I separate my filter colors and I use a nucleus stain, I have two cells, okay? And I should stop for a second and tell you, this is my composite image. All of these combined are in my composite image, but when I separate out all the channels, I have the DAPI stain there. And this DAPI stain is only for the nuclei. So you can see I have two nuclei there. When I use my markers specific for circulating tumor cells, in other words, my red triangles, I can get a very clear image of that cell being green. In addition, when I use another antibody labeled with a different color for a different marker on the circulating tumor cells, I can once again find that that particular cell is illuminated red. When I use my antibody specific for white blood cells, though, you can see that this cell is not illuminated, but this lower cell is illuminated, which tells me in this image with two cells, this one is a circulating tumor cell, and this one is a white blood cell. We were then able to take this cell and further characterize it, because although these images clearly represent that it's illuminated quite beautifully, the problem is all of them don't illuminate that well. So if our colorimetric assay is iffy, it has shades of gray, we're able to take it off and we do a FISH test, or further characterization, to determine that this cell is indeed a circulating tumor cell. What we could do, we could then take that cell right off of the filter and we could sequence it. We could look for mutations. We could find the mutations that's out there that would allow us to further characterize the cell and potentially, down in the future, determine what might be the best treatment options for this patient. Now, I've shown you the filtration process, and I don't want to make it sound like there aren't other options out there. There are many options out there by wonderful companies, by great scientists that have had tons of money placed into it. Essentially, these are the different commercially available or soon to be commercially available enrichment isolation strategies. Immunomagnetic, and we'll look at these a little bit shortly, by Cell Search and MAX, microfluid and immunomagnetic by MGH and Biosystems, electrophoresis by Silicon Biosystems, filtration by ICET and Ikonisys. None. Why enrich it? Why not use a high-powered microscope and just look at every cell? So instead of putting all of your cells onto one slide, why not put them onto 10? Put them on 10 slides, put it into a high throughput microscope, and then analyze it from there. That way you don't have to worry about any loss due to the enrichment steps. And in a recent paper out, centrifugation density. So I'm going to highlight a few of the key ones because I think they're quite interesting and wonderful to understand when you want to know about where the field of circulating tumor cells is going in the future. So now I'm going to take my antibody, and instead of putting my antibody, instead of giving it a color, I'm going to attach it to a metal sphere. Why metal? Small, round, easy to produce, but it's also magnetic. Now my antibody, bound to a metal sphere, can attach to the circulating tumor cell, and then I can use a magnet. That magnet will bind and grab all of the circulating tumor cells right out of the system. And Cell Search by Johnson & Johnson, originally by Veridex, did just that. They actually have an FDA-approved product, and this FDA-approved product uses a magnetic bead. It binds the circulating tumor cell at this given orange circular marker. It then uses this other marker, and it allows it to use a different antibody with a different color, as represented previously. And it also has these CD45 markers to allow you to recognize the white blood cells. The exciting part is that this is really the gold standard in the field right now. It is an FDA-approved product that is widely used. However, you'll see shortly, there are bigger, better things coming. The other idea, though, is instead of binding your antibody to the circulating tumor cell, why not bind it to the white blood cells? At that point in time, you can remove the red blood cells, you can remove the white blood cells, and what are you left with? You're going to be left with just your circulating tumor cell left in solution. So you take your antibody, specific for white blood cells, you attach it to a metal sphere. The metal sphere can then grab all of the white blood cells out of the system, and your magnet grabs and removes all of your white blood cells. Mass General has developed a CTC eye chip. And the exciting part about this, although untested, it's in the literature, and I'm sure they're in great studies right now, is that you take a tube of blood, you add the tube of blood right to this instrument. This instrument will allow you to take the blood going through posts, basically like walking through a maze, and it will remove the red blood cells, the platelets, and the free beads. And then it will allow, and I gotta stop for a second, you're also gonna add your antibodies for white blood cells bound to metal spheres. Okay, that's important to recognize over here. So your blood, you add your antibodies bound to metal spheres, which are specific for the white blood cells. You're able to remove the red blood cells, the platelets, the free beads, and then you're able to eventually get down to this point where, using magnetism, the white blood cells are displaced, and you're left with, coming off into this tube, just your circulating tumor cells. Now, the interesting part about this, if you think going back to what I said about the sequencing, is that if you can further characterize these circulating tumor cells by sequencing, by FISH, or other methods, you can add really vital information to the medical community. Another idea is just the enrichment technique. You take your cells, as I said before, don't do anything to it. That way you don't have to worry about loss, false binding, things like that. You can get all of your cells, you put it on a slide, you hit it with your antibodies labeled with color, and you allow it to just be scanned by a high throughput microscope. The microscope will go through it, it'll find your colors, and then you can go back, take the cell right off the slide, once again, further characterize it, or potentially do some other characterization along the way. The other interesting property here, or the other technique that I really like, is the silicon biosystems, their DEP array. It establishes a non-uniform field which exerts a polarized effect on things in the system, such as cells. That's not the important part. The important part of this is that you are going to be able to isolate and recover a pure live cell. How well it works, don't know. It's out there right now, it's new to the field, but it is extremely exciting. Imagine being able to take off a live circulating tumor cell and being able to expand it in the lab and then test it with a bunch of therapeutics. So what the field of circulating tumor cells is actually going to do, it's going to allow for detection of early cancer, right? Someone comes in, they want to know, I don't feel well let's take some blood, we'll do a test. It has the potential to detect cancer early. It has prognostic indications. In other words, I did find a circulating tumor cell, I don't know where the tumor is, what are my outcomes? What are going to be my outcomes? Well, if you're able to characterize these cells, you can certainly have something that would be quite powerful in understanding what the direction of your cancer is going to go. Certainly screening, which is what we're talking about. You can monitor disease recurrence. Let's say I have some cancer. I want to know, is it back? Has it spread? Did I get rid of all of it? Well, we can't find the tumors all the time right now. This is one method. And of course, I do want to mention personalized medicine. And the reason for that is, if you can take that cell, take a live cell, and allow it to be replicated, allow it to be tested on, you are going to have a really powerful tool in order to give therapeutic information to the medical community. Okay. Sorry.